Good morning, children. So let us continue with the sixth chapter, Molecular Basis of Inheritance. In our last video, we have learned about transcription, and today we will be learning about genetic code and mutation. So shall we start with the topic? Okay. So first, let us start with genetic code. It is the relationship between the sequence of nucleotides on mRNA and the sequence of amino acids in the polypeptide. In 1954, George Gamow a physicist proposed that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. That is triplet code. This can be explained as if one base coded for one amino acid, only 4 amino acids could be specified. That is, the protein could contain only 4 amino acids. If a sequence of 2 bases coded for one amino acid, the 4 bases could specify only 16 amino acids, which is also inadequate. But if a sequence of 3 bases coded for one amino acid, the 4 bases would specify 64 amino acids. But this generated many more codons than required. In 1960s, the proof that genetic codes are in fact triplets came from the researches of the following scientists. That is, number 1, Hargobind Khurana. Hargobind Khurana developed a chemical method for the synthesis of RNA molecule which defined base combinations, homopolymers and copolymers. Number 2, Marshall Nirenberg. He put forward a cell-free system for protein synthesis that helped in deciphering the code. And number 3, Severo Ochoa showed that the polynucleotide phosphorylase also helped in polymerizing RNA with defined sequences in a template-independent manner. On the basis of the discoveries, the spellings of the genetic code were put together in a checkerboard as given here. Can you see? This is here is the first position of the triplet. This is the second position of the triplet, and this one is the third position of the triplet. And using this, the 64 different codons were formed. You can see 64 codes were formed which expresses for different types of amino acids. All these are given. Now, out of the 64, 3 were stop codons, that is UAA, UAG and UGA. These are the stop codons. Clear? The various features of genetic code which can be explained by using the given checkerboard are: number 1, codon is a triplet. Out of 64 codons, 61 codes for 20 amino acids and 3 codons, that is UAA, UGA and UAG, do not code for any amino acids. Thus, they function as terminating or stop codons. Next is genetic code is unambiguous and specific. Unambiguous means it has only one interpretation. Thus, one codon codes for only one amino acid. Example, CCU codes only for proline and not for any other amino acid. Next one is, since the number of codons is greater, all of the amino acids which with exceptions of methionine, that is AUG, and tryptophan, that is UGG, are coded by more than one codon, a feature referred to as the degeneracy of genetic code. Example, GGU, GGC, GGA and GGC all specify the same amino acid glycine. You can check in the checkerboard. The codon is read in mRNA in a contiguous fashion. Contiguous means touching, that is without any punctuation. Thus the code is comma-less. Then the genetic code is nearly universal, that is particular codon codes for the same amino acid in all organisms except in mitochondrial codons and few protozoans. AUG is a codon with dual functions. It codes for the amino acid methionine and also acts as an initiator codon. So children, now let us study about mutation. The sudden inheritable change in the genetic material is defined as mutation. Sudden, immediate, okay. Inheritable means they are transmitted from parents to their offsprings. So the sudden changes which can be transmitted from parents to their offsprings in the genetic material is called a mutation. And it is of different types like point mutation, mutation in a single base pair which is replaced by another base pair. Example, in sickle cell anemia, a point mutation in beta globin chain results in the change of amino acid residue glutamate to valine. Okay, because of change in only a single base pair, a new amino acid is produced. That is called point mutation. The second one is the frame shift mutation. It is the change in the reading frame because of insertion or deletion of base pairs. Okay, there will be a change in the frame. Why? Because some of the base pairs are inserted or some of the base pairs are deleted. For example, let us take an example. This is the sequence A, T, C, G, C, T, T. Right? Now, if we remove this T and this C, okay, that is, there is deletion of base pairs, then the sequence will be A, C, G, T, T. Right? Earlier the genetic codes were ATG, GCT. Okay, now it is ACG, TT, whatever is there further after this. Okay, so the genetic code changes, the complete frame changes because of the deletion of base pair. Similarly, if we insert some of the base pairs, suppose here T is inserted, here also T is inserted, right? So the new the new frame which will be produced will be A, T, T, C, G, C, T, T, T. Okay, so again the frame changes which leads to the formation of a different type of amino acid. So this is called frame shift mutation. Okay, then Insertion, as I've explained you, if one or more nucleotides are added in the DNA segment, it is called insertion. If 3 or its multiple bases are added, they will not change the reading frame. In fact, they will form a protein with new amino acid. And what happens in deletion is, if one or more nucleotides are removed from the DNA segment, it is called deletion. Here also, if 3 or its multiple bases are removed, it will not change the reading frame but will remove one or more amino acid from the polypeptide. And last one is the silent mutation, where there will be no change in the, there will be addition, there will be insertion or deletion of the base pairs, but it will not result in the change in the polypeptide chain. Okay, so children, that is all for this video. In our next video, we will start with the new topic, that is translation, that is how the proteins are produced from mRNA. I hope this is all clear to everyone. Thank you.